Well, good morning, everyone. Nice to see a good turnout. Welcome to the UK Cereal Pathogen Virulence Survey stakeholder meeting. It's good that we've got a face-to-face meeting this year, which is always, I think, more interesting and interactive than the online version. My name's John Lucas. I'm chair of the committee and has the job really of giving you an introduction and overview of the day. So with no further ado, we'll carry on. Okay, so of course the core focus of the Virulence Survey is disease surveillance, and I think we all know the importance of disease surveillance, especially after recent years and the pandemic and all the rest. So just a few slides indicating the diseases that we're most concerned with. This one, yellow rust, is of course a core focus of the main survey. Yellow rust to me is an interesting disease because when I first started to work in this area, it was seen as a relatively uniform pathogen in terms of its genetics. Most of the epidemics were clonal, and once you'd identified the principal cause of the disease, there wasn't too much diversity to cope with. This has all changed in recent years with new strains of the rust fungus coming into the UK and Europe, and we now have a sexual cycle operating, and this has created much more diversity and has made the disease surveillance, if anything, more important because we're dealing with a quite complex situation. This is actually a slide from John West showing early stages of a stem rust epidemic. This one was actually part of his research where these disease foci appearing in the field originated from a single plant that in May the epidemic started to spread. So under favourable conditions, yellow rust can spread very quickly, and of course if we've got advanced intelligence in terms of the strain of the pathogen that's involved or the strains, it's helpful in terms of understanding variety susceptibility and also probably the need for fungicides as necessary. Now, of course, that's not the only problem that the farmer has to deal with. Here's some others, and in the survey we include brown rust and mildew. Septoria is a focus because of its importance in the wheat crop in particular, and last year, for instance, there was a severe Septoria year. At the moment, there's not a lot of analysis of the populations. It's a complex disease with many different strains in the population, but it is, of course, one of the principal problems for the farmer. So we have a Septoria watch function, which is if anything new is happening, if a variety, for instance, is breaking down, it's important to try and get that information out. Now, in terms of programme today, we've diversified into some other problems that have to be dealt with. Barley yellow dwarf virus, here symptoms on wheat. We have a presentation from Lawrence Bramham on that. I think I have to point it this way. And actually, this is a disease that I don't think we've discussed previously at these meetings, but we have 3 presentations on ergot, which is primarily caused by Claviceps purpurea, although there are other species in other parts of the world. It's quite common on grasses and wild grasses, and of course can infect cereals, and the concern in this case is not direct effects on yield, it's to do with the contamination of the food chain by ergot alkaloids. Now, the picture on the right there, when I was a student, this book was published called The Day of St Anthony's Fire, and it reads a bit like a detective novel actually, but it's about a real event that took place in France in 1951, I think it was, where unexpectedly a lot of the residents of one particular village started to suffer hallucinations and behave very strangely, and this was initially not understood to be anything to do with ergot. In fact, mercury poisoning was considered as a possible cause, but eventually they traced it all back to ergot infected flour used in the local bakery. I think that book's still out there somewhere. On Amazon I could see there was a few copies. I think it's out of print, but I think you can possibly get hold of it. The picture here, of course, is the sclerotia formed in the flowering head, and there's the problem. If those sclerotia get into the grain, that's where toxicity can occur in the food chain. Quick mention then of what tools do we have available for dealing with these diseases. Obviously there are some, such as sowing date, which can be important, particularly perhaps with the virus infections there. That's relevant to preventing spread into the crop too early. Probably the cornerstone of control, and obviously the thing that we're very focused on, is resistant varieties. Fungicides, of course, are available for situations where the diseases are likely to be a problem year. Biocontrol, I put a question mark there. There's a lot of interest in it now, using biological agents to control disease, but probably in broadacre crops at the moment it's not a major focus, whereas in glasshouse crops, for instance, there are many examples of successful biocontrol strategies. Now, of course, we've got these tools, but there's also the constraint of pathogen variation. And pathogen evolution for both virulence towards our resistant varieties and resistance to the fungicides we can use are two of the main issues with why disease is still a continuing problem. So I put this slide in every year, actually. It shows the variety ratings in the recommended list, and encouragingly, if you see these are the ratings at the bottom, so those are the colours of the columns. This is starting in 2020 and moving on to the 25, 26 list. I think the most important thing is showing that the breeders have been doing a very good job here in terms of yellow rust, in that most of the varieties are moving towards the right-hand side, which are the higher resistance scores. So good progress is being made on that front. Of course, these resistances can still be compromised by the emergence of new races of the pathogen. So it's an ongoing business of keeping up with the epidemics. Very quick mention of fungicides. These are two slides. This is a slide kindly given to me by Nicola Hawkins at NIAB. What this shows actually is prothiaconazole on the left-hand side here, and this is Septoria and azole sensitivity. And you can see, actually, this is in terms of sensitivity, you can see that as the years go by, the population is shifting to the right. And that is gradual emergence of less sensitive isolates as time goes by. So that's prothiaconazole, which was launched in about 2005, I think it was, or possibly just a bit before then. This is the more recent compound, which I think of as Revisol. I can never quite pronounce the azole name. And I think you can see there's obviously some variation there, but you haven't yet got that more progressive emergence of resistance. Nonetheless, this all needs keeping an eye on to understand the most efficient use of chemical control. And this is the SDHIs, just two examples here. And again, you can see that there's been some change in sensitivity over time. But it's a much more complex picture, this, because there's a whole series of different mutations involved, and some of them compromise sensitivity and others seem to have a minor effect. So at the moment the SDHIs are still useful tools, but we have to keep an eye on it to make sure that they don't go the way of the strobilurins, for instance, QOIs. Okay, so here's the programme. I'm not going to read all these individual bits to it, but obviously that's my introduction. Then we're going to have the Virulence survey reports, both from the 2024 season and the winter wheat trials, and then on to these other diseases, which I'm looking forward to because, as I said, we haven't really had presentations on these before in this particular meeting. We're going to have a break round about 11:15, so the first part is really the presentations on what has been done in the survey last year. Okay, I think I can hand over now to Kostya. Sorry?
Combination of both. It's myself and Kostya.
Oh, yeah. Oh, okay. I do beg your pardon.
No, you're all right.
Okay, so I should really introduce our speakers. Kostya's head of plant pathology at NIAB, where he leads strategic, applied and commercial research, encompassing biology, detection, surveillance and management of diseases of field crops. He was previously actually at Rothamsted, where we worked together actually for a while, and he led a variety of projects focused on resistance to viral and fungal diseases in barley and wheat in particular. He's now the current lead of the UKCPVS project. Hugh is a cereal pathology specialist at NIAB, and he's the new technical lead for the UKCPVS, following actually the retirement of Amelia Hubbard, who some of you I'm sure would have known. He's previously worked on a variety of broadleaf and cereal pathogens at NIAB before focusing on rusts and the Virulence survey. So sorry, Hugh, I should have introduced you first. Okay, over to you.
Okay, good. Oh, that was quite loud. Good morning, everyone. My name's Hugh Davis, and welcome to the UKCPVS steering stakeholder meeting. So I'm just going to go over some of the data that we've had this year. So sort of a step-by-step how the presentation from myself and Kostya are going to go today. So start off with a bit of an introduction, which some of you may already know about, but I'm just going to go over for those who don't. And then we'll talk about the wheat yellow rust and brown rust results, a bit about the sampling, and then sort of how the UKCF is going to change from 2025 up until 27. So just a bit of background about the project. It was founded in 1967 after an outbreak of yellow rust on the previously resistant variety of wheat called Rothwell perdix. The UKCPVS is there to identify changes in pathogen populations or any new races that we may be detecting in the UK and how this might affect UK agriculture. It relies heavily on farmers, agronomists, trial staff, breeders, researchers, all sending in their samples for the study. So every sample you send in, we'll process and hopefully, we might be able to test it. It's funded by the AHDB and there's a contribution from AFRA as well. So the three main pathogens that the UKCPVS looks into is wheat yellow rust, wheat brown rust and barley powdery mildew. And this has changed over the years. When it started, it focused on Rhynchosporium, the rusts, Septoria and some crown rust and some other diseases as well. So from 2023 to 2024, we had a couple of changes to the project. So we stopped running adult plant trials in 2024, but we put the focus on the seedling stage growth room studies for yellow and brown rust. We introduced a new differential set, which was reduced, and this was used for our pathotyping of yellow rust and brown rust. We tested more isolates on the full set of the recommended list and the candidate varieties, and where it's possible, we did try and improve our in-season reporting. For wheat and barley powdery mildew, for wheat, we didn't see any sort of unusual sightings or reports in 2024. And although in 23 there was some susceptibility seen up in Scotland for barley powdery mildew, we didn't actually receive any samples this year or last year. So this is sort of a process of how we deal with your samples. So when you send us in a sample through the post, we will receive it. We'll send you an email with the isolate code and just acknowledging that we have received that sample. And then this gets damp chambered overnight. And for yellow rust, we will isolate a single yellow rust stripe. And then this gets put onto a pot of susceptible wheat and then grown up in the growth rooms. And sort of about 10 to 14 days later, you'll see some sporulation. And then from there, we can collect spores, and that goes into our long-term storage. Brown rust, it's slightly different. We'll damp chamber again overnight, but we'll take a single pustule off the leaf sample that you've sent us, and then this gets put onto a detached leaf agar box, and then after about 10 days, you'll see sporulation, and then those leaves get put onto a Burkard pot in the growth rooms of a susceptible variety to brown rust, and again, sort of 10 to 14 days later, you'll see sporulation, and we can collect the spores. And then from all the samples that we receive, we'll select 25 yellow rust and 10 brown rust isolates, and this is based on geographical location or the variety. So if you send us something that's got a rating of 9, we're definitely going to be interested in trying to test that one. These then get tested on the full set of recommended list and candidate varieties, as well as the differential lines. So this is sort of timeline of how we deal with it within the year. So in summer, perhaps starting in March up until late July, maybe early August, we'll be receiving samples. And then, as I said, we will select the 25 yellow rust and 10 brown rust, and this will go on to the testing. So in late summer, autumn, perhaps a little bit into sort of November, we'll be doing the differential tests. So this is to understand the pathotype of all the isolates that we've selected. And then these also get the same isolates then get tested on the recommended list and candidate seedling tests in the growth rooms again. And then this, the results we get for the recommended list and candidate varieties, you'll see this in the recommended list young plant rating in the RL booklet. So this is just a bit of sort of background about the differential tests. So that's to get the virulence profile, which we sometimes call the pathotype. And that's basically just a list of all the virulence genes each isolate carries and any additional test cultivars. So in this table here, on the left column, you've got the differential cultivars, and on the right, you have the resistant genes that correspond to it. So if a isolate, for example, avocet 1, if an isolate infects avocet 1, then it's said to have the virulence gene YR1. And by the time you've done the full test and you've got all the scores, you'll get a virulence profile that looks a little bit like this. So you have the start where you've got the resistant genes, and then the end where it's the additional cultivars. So you've got rendezvous there with RE, Solstice, Cadenza, Apache, and so on. So now for wheat yellow rust for 2024. We received 68 samples in 2024 from 17 different counties, and we also had one unknown county. We saw that the weather was quite conducive for rust this year, so we had that nice mild winter, and then it was followed by quite a wet spring, so machinery is struggling to get onto the fields to give the fungicide applications that were needed to prevent some of the rust. This pie chart here just shows you the sort of breakdown of the varieties that we had sampled from. So KWS we had 15 samples from, Crusoe we had 6, and then Gleam, Graham, Skyfall, and LG Red Bold we had 5 samples from. You can take this with a little pinch of salt because KWS is one of the more commonly grown varieties in the UK, so you'll probably expect to see some more samples from that one anyway. This is the geographical location. So the graphic on the right, there we go, shows you a map of the UK, and anywhere that's coloured in, we've received samples from, and the darker the colour, the more samples we've received from that location. The pie chart, it shows you a breakdown of sort of where they were from. So we received 41% of our isolates from Lincolnshire, and then we received 15 from, I don't think can I go back, please? Thank you. 13 from Norfolk and 12 from Cambridgeshire. So we have a quite good distribution this year, but obviously predominantly from Lincolnshire. So these are the 25 isolates that we selected for testing, and you can see all the details, so where they've come from, the host variety, and in the far right column you've got the seedling virulence profile. So if anyone sent a sample in here and we replied with you've got sample 24014, then you can see your virulence profile for that isolate there. At the bottom, at 26, we also tested a mixture of 3 isolates. So we selected 24003, 4 and 7, and this was to compare how using a mixed isolate compares to just individually testing them. So we found that the resistant isolates and the susceptible ones are easy to pull out just using a mixture there. So this graph here shows you a 5-year comparison of the differentials and using the yellow rust genes. So you can see the percentage of isolates that are carrying the virulence to the yellow rust genes here. So we didn't detect any virulence for YR5, 10, 15, and there was little to no virulence for YR8 this year. You'll also see some sort of fluctuation in the scores from perhaps like the YR1, 17 and 13, which have been quite consistent over the years, but we're just putting this down to some scoring differences between myself and Amelia, but this is quite easy reflectable, but the main messages remain the same. Thankfully, there's no new varieties coming out with 5, 10 and 15. This one is just showing you the additional cultivars with the unnamed resistant genes. So again, the percentage of isolates carrying that virulence. We found that the percentage of isolates carrying virulence for ambition increased this year, while all the rest reduced, but the additional cultivars are known to vary quite a lot in there with the isolates carrying virulence, so that's quite normal to see this sort of fluctuation year on year. So now the same 25 isolates that we selected for the differential tests get tested on the recommended list and the candidate varieties. This table here just shows you their reactions. So anything with an R was resistant and anything with an S was susceptible, and the number next to it just corresponds with how many isolates that was susceptible to. So we found 14 of the recommended list and 15 of the candidate varieties were susceptible to up to 16 of the 25 isolates we selected, or 64%. We saw that Graham, that has a rating of 7, infected 32% of isolates, and LG Redbold, KWS X-Days, infected 24, again with a rating of 7. And the candidate variety, Arnie, slight error there, I'm afraid. It should be the recommended list variety, KWS Zayat, was susceptible to 64% of isolates. All the recommended list varieties with a rating of 8 or 9 performed really well at the seedling stage based on their ratings, so that's positive thought there. So the data from our seedling test you can find in the RL booklet here in the column that I've highlighted in yellow. So it's under the yellow rust young plant data, and that's indicated with the R or S. It's going to go through there. So these are the ratings for 2025-26 with our results from this year. So anything with an S is susceptible, anything with R resistant. An interesting one to point is for, say, RGT Goldfinch, which is a new one to the recommended list for 25-26, with a rating of 9, has been put down as susceptible because it was susceptible to 8% of the isolates, whereas you take KWS diet, which was susceptible to 64% of isolates, was also indicated by an S. So those varieties that are susceptible to multiple isolates, monitor these, and if they need any applications, do just get them in the field. So this just highlights once again those varieties that were susceptible and resistant. So anything in blue was your recommended list, anything in green is a candidate, and everything in the red box was susceptible, and everything in the green was your resistant isolates. So just to summarise the wheat yellow rust for 2024, we received 68 samples from 17 counties and 25 different varieties. We didn't see any major variety breakdowns this year, which is great, and no virulence was detected for YR5, 8, 10 and 15. The percentage of virulence for additional cultivars continues to fluctuate year on year, and 17 of the recommended list varieties and 8 of the candidate varieties were resistant at the seedling stage. We found that 14 at the recommended list and 15 of the candidate varieties was susceptible to up to 16 out of the 25 isolates we selected for testing. And as I mentioned, the mild, wet winter was quite conducive for rust development this year, and continue to monitor those varieties that were susceptible to multiple isolates at the young plant stage. So now going on to brown rust. We received 65 samples this year, which is more than we usually do, from 10 counties and 31 different varieties. And again, weather conducive for brown rust this year. We had quite an early epidemic, this sort of starting in March, April, and then we had a really high amount recorded in July. The most sampled variety was Crusoe, and then followed by Champion with 5. This is the geographical locations of where we received all the samples from. So 29 of the samples came from Lincolnshire, and 16 from Norfolk, and 11 from Cambridgeshire. So in addition to the 25 yellow rust isolates that we tested, we've selected 10 of the brown rust, and these are the 10 that we selected. So again, you can see which variety they came from, the isolate code, and the location. And in the far right column, you've got the pathotype of that particular isolate. So similar to the yellow rust slides, these are some graphs just showing you the changes over the past 5 years. So this is for the leaf rust genes, and now leaf rusts fluctuate just like the additional cultivars. The leaf rust genes do fluctuate year on year. We didn't detect any isolates with virulence for LR2A or 2B, which is great because that's matches previous years. And this is for your additional cultivars with your unnamed R genes. So we had a fixed virulence for Glasgow, KWS Firefly, and Robigus. And then again, just some fluctuation within the others. This could be down to my scoring and some of the normal fluctuations year on year. So this table on the right shows you the recommended list in blue and the candidates in green. So only one variety was resistant to all the 10 isolates, which was RGG Goldfinch, which is a candidate variety. And all the RL and most of the candidate varieties was susceptible to at least 3, but up to 10 of the isolates. SY Cheer, KWS Diet, Graham, KWS Solitaire, Bestie, and Rufus was susceptible to all 10 of the isolates. Skyfall, which had a RL rating of 9, was susceptible to 4 out of the 10 isolates that we tested. So this table just sort of breaks down on some of the RL varieties and their brown rust ratings against the 10 isolates that we selected for testing. So if you take Crusoe, for example, with a rating of 3, it was susceptible to 7 out of the 10 isolates we selected, and that was 24, 01, 3, 9, 13, 15, 19, and 30. But it's quite a good mix of which isolates they're susceptible to, but you can see 24, 0, 5, 1 wasn't a particularly virulent isolate. So Here again, just sort of summarises which were susceptible, which were resistant. So everything in blue was your RL, and everything in green is candidate. So only RGT Goldfinch was the only one that was resistant to all 10. But we did also see Diamond, which is another candidate variety, was resistant to 9 out of 10 of isolates we tested. So it was only one that broke through. So to summarise the brown rust results for 2024, that should be 24, ignore that. We received 65 samples from 11 counties and 31 different varieties. The most sampled variety was from Crusoe, and we selected 10 isolates to test on the differentials and the recommended list and candidate varieties. We found virulence for the leaf rust genes LR1, 3Ka, 10, 13, 14A, 15, 16, 17, 26, 28, 37. And no virulence was detected for leaf rust genes 2A and 2B. A single isolate from Lincolnshire, we found the virulence for STIG, which carries the gene LR24. And all the candidates and pretty much most of the, all the RLs and most of the candidates were susceptible, apart from RGT Goldfinch, which is resistant to all 10, and Diamond resistant to 9 out of 10. Okay, we'll go on to the next part. So, sampling for this year.
I will stay here.
Okay. So how are your samples used every year? So you'll send them in, we'll make isolates out of them, they'll go into storage. But how else are they used? So they could be selected for future work for the recommended list and the variety testing. So they might be the isolates that we select to use on our inoculated trials. Obviously, they are tested in the growth rooms at our seedling stage resistance, and breeders can use these to screen breeding lines, which is good for future commercial variety resistance. We can also use these isolates for testing ag-chem or biocontrol research and testing new products that may help you out in the field at some point. They're also frequently used for research studies, say PhDs, any different collaborations with other organisations. And where it's possible, we try and participate with international projects. And we always keep a number of ampules in our long-term storage here at NIAB. So if you're sending in a sample, we'd like you to use one of these sampling sheets. So you can find this on the NIAB website if you look for the UKCPVS. And if you could, just put as much detail on there as possible. So the crop, variety, date, any fungicide applications, or the percentage of disease. Anything you can tell us, basically, it helps us out for just registering that sample. So with your sample, you'll put that in the envelope and the sampling sheet, and then put it into a paper envelope, no plastic, please. And then use the words Free Post UKCPVS on the front. If you write anything else, this may get held back by the Royal Mail, and then by the time we get the sample, it might not be the best sample to process this from, so we might not be able to get an isolate from that. So no plastic bags, paper envelopes, and only Free Post UKCPVS on the envelope, and then we're off to a great start. So if you could send us in a sample this year, that'd be really, really helpful. And yeah, thank you. So now I'm going to pass you over to Kostya. He's going to talk you through changes to the project and how it's going to work for 2025 to 2027. Thank you very much.
Thank you very much, Hugh.
Welcome.
So hello everyone. My name is Kostya Kanyuka. I'm head of pathology here at NIAB, but also been leading UKCPVS since about April last year, when Charlotte Nellist went on to a maternity leave. So I enjoy working with you and the new team in UKCPVS. So I must say that UKCPVS is probably the longest running project known to man, at least in the UK, I would say. And this project was running from NIAB for, I think, 57 years, if I remember correctly. So last year, AHDB decided to actually retender this project. And I'm glad to say that the project will stay with NIAB for at least another 2 years, possibly another 5 years, because there is a possibility to extend this project, I believe, for subsequent years. Oops, I've pressed the wrong button again. Which is the right button to press? The central one or not?
Yes, the one on the right.
Okay. So because of the much reduced funding available, I think, for this project from next year, and also because of the high emphasis required for producing the outputs of relevance to stakeholders and levy players, and much less emphasis on the science, the UKCPVS in the next years will look very different, unfortunately. But we are hoping to still be able to deliver very useful outputs for you guys. So the new project will have 3 work packages listed here. So work package 1 is support breeding for durable resistance. Work package 2 is focusing on the young plant resistance status of recommended list and candidate varieties. And in work package 3, it's basically just maintaining our entire collection of isolates and also for the differential lines for wheat and other species. So the key objectives of the projects are slightly different to the previous year. So we will collect and characterize the virulence profiles for wheat yellow rust and brown rust isolates from diverse geographical regions and different wheat varieties. We determine the resistance status of recommended list and candidate varieties specifically for wheat yellow rust at the young plant stage exclusively. Again, we are not funded to do the adult plant resistance, and this data from the young plant stage will go into the RL booklet. We will maintain cereal rust isolates and the corresponding differential wheat lines, as I mentioned. And then we will also, responsibility in this project will be to bulk up and provide selected isolates for inoculated RL and VL disease trials for different operators. Not sure why it's not moving. Okay. So a little bit more about work package 1, which is supporting breeding for durable resistance. So 1.1, objective 1.1 is to sample with yellow rust and brown rust varieties. We aim to sample from about 50 to 100, to receive 50 to 100 different samples for those isolates, for those pathogens throughout the season. So we will focus on collection from as many geographically diverse areas as possible, and from as many different wheat varieties as possible as well. But we will have a priority, we will focus on varieties with high resistance ratings at the adult plant stage from the booklet, from the RL booklet. To be able to sample as many isolates or to receive as many samples as possible, we will reach out to AHDB RL trial operators, to DEFRA Pathogen Disease Survey project, and also to NIAB agronomy members. And we will keep maintaining, generating the informative maps and pie charts and provide them to AHDB and then YHDB to levy players, I believe. And then under the same work package, we will isolate with yellow rust from 21 samples that we receive and 10 isolates for wheat brown rust. We will have to pick, because we will have over maybe 100 samples in the collection. We will have to pick isolates that are fairly representing the source varieties and also their market share. And those isolates will be then tested against a completely new differential sets for with yellow rust and with brown rust to reflect the importance of delivery for the levy players. And I will touch on that on the next slide. We will also keep providing isolates for VL and NL trials. And the idea is that we provide 3 isolates that would jointly cover the widest possible range of virulences for testing. It's a bit slow. I don't know what's going on. Right, so new differential sets. So for wheat yellow rust, we decided to include varieties that carry these 4 genes, 5, 10, yellow rust 5, 10, 15, and 24. And the reason for that is because up so far, we have not seen any virulence in the population towards these genes. So it is very important to monitor the population and see whether there is a breakdown in the future. While 8, there is a very low frequency of virulence, and again, we thought it will be important to monitor whether this will increase in the future. We also decided to include these different 4 or 5 different varieties. They would help to infer common genetic groups, because we don't do genotyping anymore, but those varieties could be indirectly used to predict to which genetic group the isolate belongs, not just the pathotype, but the genetic group. And the rest of the differential set will comprise 17 recommended list varieties with the highest disease-resistant rating. And again, I think it would provide useful data for growers. If we see any breakdown of resistance in the most highly rated varieties, I think that will be important. And the way the screen will be carried out this year is, instead of using individual isolates, we will use mixtures of 3 isolates. And then if we see any breakdown or any virulence in the mixture towards particular resistant gene, we will rescreen the isolates individually. We actually tested, and I'm not sure, I think we will not mention in the first part of the presentation, but this year in 2024 season, we actually tested the possibility of using a mixture of isolates instead of using individual isolates, and it worked fairly well for our purpose at least. And for the wheat brown rust isolates, again, we will include the 3 genes mentioned on the top row, because we know that they're present in UK varieties, but the frequency of virulence remains very low. And we would like to see whether this will be changed in the future or not. LR20 and LR23 virulence we detect occasionally in the UKCPVS, but it's not yet fixed. So we also include those lines carrying those resistant genes in the differential set. And finally, we also have the LR, 2A, 2B, and 2C genes, plus RGT goldfinch. And from Hugh's presentation, you would learn that this particular variety up so far has been resistant to all the isolates that we've tested in UKCPVS for the brown rust. So if there is breakdown in RGT goldfinch, I guess you would like to know as well. And also, more recently, we acquired additional lines, additional wheat lines, carried more newly identified resistant genes named from LR38 to LR37, and those will be also included in the screen. And screening those lines will tell breeders whether there are any useful genes that could be introduced in the breeding program to control brown rust. Now, moving on to young plant resistance status of varieties. So we will use our isolate collection to screen up to 60 recommended list and candidate varieties. Actually, there are about 60, I think, on the list right now. And again, a mixture of 3 isolates will be used, the same isolates that we will provide you for RL and VL trials, and the same isolates that will go into the young seedling stage field trials. So format of those trials will be 3 replicated 2-row 1 meter long plots. We will have also spread of wheat variety. From Hugh's talk, you will know that KWS Zayat is particularly susceptible to yellow rust at the seedling stage, so we will use it as a spreader. And there will be 2 rows of this variety, and then outside of those 2 spreader row varieties, we will sow the RLs and candidates. We will also have funding for the optional irrigation. This is in case the spring becomes very, very dry. So this is just to encourage, again, yellow rust infection. And we will try to drill those field experiments in February, March, if the weather permitting. But probably beginning of April we'll also do, I think, for our purpose, because yellow rust needs to be exposed to low temperature for germination and also for plant infection. And we will score our plots at several time post-inoculation using at least 2 different methods. And you can go and have a look at the AHDB website, how we've done it last year. So, and in parallel, the same isolates will be screened also under growth room conditions here at NIAB, and we will use the same mixture of 3 isolates. And the reason for that is that in case of our, for example, field experiment is destroyed for whatever reason by animals or humans or due to the weather, maybe yellow rust will not take up very well, we will have at least the data from the growth room experiment. And it would be also nice to correlate both sets of data. Next slide, please. Okay, work package 3. This is the smallest work package. Basically, oops, gone too far. Yeah. It's just maintaining our cereal rust isolates and maintaining differential lines. For the cereal rust isolates, we normally would enter them into our database, and we store all new isolates together with the historic collection in our 4-degree in the cold room, basically, or in the fridges as vacuum-dried spores in the sealed glass ampules. And I think the breeders who receive samples would know the format because we supplied them with the glass ampules of the rust isolates. We will keep updating the database. We will also store the newly purified isolates similar to the historic collection. And then we have a capacity to provide industry or researchers also with all the isolates of wheat, barley, oat, rice, if they're interested to. Regarding the wheat differential lines, we will maintain all the original differential sets in our long-term seed store, but also next year we will multiply up to 50 differential wheat lines during in 1 by 1 meter field plots. And then if these materials become available in the 26-27 season, we can actually include differential set in our young plant trial as well. All the lines will be harvested by hand from the central kind of portion of the plot to ensure that we have a pure seed. There is no seed contamination. And then, yeah, as I said, those seeds or those lines could be included in 2026-27 trial. So that was it about the future UKCPVS. And just, I think this is probably the final slide, just a few take-home messages from Hugh's presentation, from my presentation, firstly about yellow rust. So what we found that the fungal population, they still remain very diverse. So they're not clonal, they're still very diverse. But we haven't seen any major varietal breakdowns. And about half of the recommended list and candidate varieties are resistant to all the isolates that we test, and we test 25. So I think this is the good news. And we found no virulence so far for lines that carry yellow rust 5, 8, 10, and 15 resistant genes. And I think the breeders may decide to use those genes to introduce in their programs because they seem to provide very good resistance to the UK population of yellow rust. And finally, the highest percentage of virulence is detected for KWS Zayat, and I think the current rating is 3. It's not 4. I think it's 3. And it's also fully susceptible at the seedling stage. And this is the reason we also use it as a spreader variety. For brown rust, unfortunately, all varieties, with one exception, were found to be susceptible at the seedling stage. And I think this is important because if we have many wheat cultivars infected at the young seedling stage, this will create a problem for future epidemics. And this is what happened last year when the year was particularly good, let's say, for brown rust. We've seen a massive epidemics in the later on in the season because there was plenty of rust available around. RGT Goldfinch, rated 9. Right now, it's actually been moved from candidate to recommended list varieties in this year. It was fully resistant to all 10 isolates, so I think it's an excellent variety for control of brown rust at the seedling stage. And virulence for LR24 is actually fluctuated. It was on the rise in 2023, but last year we found it kind of got down to a basic level of about 10%, so there is not a lot to worry about, I think, at this stage. And finally, a few other points is please refer to AHDB recommended list for resistant ratings, which include also unplanned resistance, and also AHDB watch list when making your variety choices. And pathogen populations are dynamic and diverse, so keep an eye on your varieties, even at the young plant, the seedling stage, and apply spray if you think it's necessary. And for UKCPVS, please monitor any unusual sightings and send us sample. And I just wanted to emphasize, please do not write anything else on the envelope apart from UKCPVS. Free post UKCPVS. If you add anything else, unfortunately, Rowan Mail will invalidate that, and we will have to pay. We'll have to go to post office, pay, and then retrieve the samples, and it takes a long time quite often. They're very cheeky. Anyway, so yeah, but please don't do that. And I think that's it, probably. Yeah, just for further information, please take a look at those links on the AHDB website for our annual reports. So the next one will be, we will be submitting actually later on today, so it will become hopefully soon available for everyone to see. And for recommended lists and press releases, sampling, and take a look at other websites, and also Global Rust Reference Center website could be also very useful. So this is the Global Rust Reference Center, operates in Denmark, and they survey yellow rust, brown rust isolates actually across Europe and probably even more widely. And we are trying to coordinate our kind of activities here in the UK and in Europe via the Global Rust Reference Center. So we have good links with them. And Leslie was helpful in establishing that connection too, or re-establishing the connection. So finally, I would like to acknowledge all the people involved in producing this data. And I must say that there was a huge change this year in terms of the personnel. So I'm new in the project. Hugh is actually new. He's never been working for UKCPVS before, but he's been fully trained by Amelia before she left NIAB. So he assumed this role, and he's doing a great job. We also have Adam Donaldson and Akhil Bonaparte, who are new to the team, so they also contributed to this survey. Janet Adams is our technician, so she's been around in this project for quite a while and helps with our growth room-based experiments. Leslie, her advice was very valuable during the season. I'd like to also thank field trials team, Charlotte, who's actually helped me to run this project successfully. She should return to NIAB to work in April this year. And also Amelia and Megan, sorry they're in different color, but they've done a massive work in the previous years, and also in the beginning of the season. I would like to thank them, Amelia. And finally, many thanks to all the people who send us samples, whether they're from NIAB or from other organizations. Thank you very much. And finally, AHDB and NAFA for providing continuous funding. So thank you very much.
Kostya. At this point, we're moving on to Sacha White. Just a brief introduction. Sacha's had about 20 years' experience with entomological research in arable and horticultural crops. He joined AHDB as a lead crop protection scientist in 2024. He was principal research consultant at ADAS and led their arable entomology group. Sacha's work focuses on integrated pest management, including work on major insect pests of cereals and oilseeds. I think that's
Thank you, John.
enough introduction. Nice to see you.
Nice to see you.
Sacha's going to be talking about spring-sown wheat trial testing young plant resistance.
So clearly I've got a background in entomology. You might be wondering why I'm talking today. Catherine Harris, I'm really presenting Catherine Harris's work, and she's on maternity leave. So this is all her brainchild, really. But I'm going to be presenting results from a pilot study looking at sowing winter wheat in the spring to test for young plant resistance to yellow rust. So resistance to yellow rust operates differently, or can operate differently, depending on the growth stage of the plant. So you can get young plant or seedling resistance, and then you can also get adult plant resistance, which usually kicks in around growth stage 55, although there is a little bit of variability there, so that can depend on the resistance mechanism and the environmental conditions as well. Now, for the what this means is that a variety that is resistant at the adult plant stage might actually be susceptible at the young plant stage. And historically, the RL has shown adult plant resistance. But knowing whether a plant is susceptible or resistant at the young plant stage is important because growers and agronomists are having to make decisions about spraying for yellow rust during that young plant stage. And so knowing this information can help them know whether it can confirm a decision to spray, or can give them the confidence to think, oh, well, might hold off a spray if they know that adult plant resistance is going to be kicking in imminently. So in the 2022-2023 RL, for the first time, there was information on young plant resistance in yellow rust, and you can see it in the disease resistance section there under mildew and yellow rust. And as Hugh was saying, this is classified as either, at the moment, as either being susceptible or resistant in the RL. So that's the S and R's indicate. So the classification of young plant resistance is based initially on the UKCPVS growth room experiments that Hugh was describing there. And here we've got the table from the 2022-2023 RL, just to take you through how the process works. So here we've got the varieties ordered by the UKFM group, and then you've also got the adult resistance rating, the numerical value, and then its adult plant categorization of being susceptible or resistant. And the final two columns show the young plant resistance as classified from the UKCPVS growth room experiments. And that was in 2020 and 2021, which fed into that edition of the RL. So those UKCPVS experiments are growth room experiments, they are seedling experiments, and they are tested with 5 isolates that are characterised as part of the UKCPVS. And as Hugh was saying, if they are susceptible, if a variety is susceptible to any one of those varieties, then it's classified as being as susceptible. Now, because the isolates that are used in those growth room experiments are a subset of the wider population of isolates that might be experienced in the field, then the performance of a variety that is classified as susceptible in the growth room experiments, it might actually vary in the field because there's a wider range of isolates out there, and also environmental conditions can also affect the activity of that resistance mechanism as well. So for that reason, we validate the growth room experiments using data from the RLs. So that's data where disease symptoms are collected prior to growth stage 55. And in 2022, there were 4 RL trials where this data was collected, so 3 in Norfolk and 1 in Sunderland. And I've anonymised the varieties, and also I've taken out the data, but wherever there's a red square next to a variety, that means that that variety had more than 5% disease, and that's the threshold. So if a variety has more than 5% disease, it's classified as being susceptible. That's the threshold that we use. And then the final column over on the right, yes, the final column over the right is the classification of young plant resistance based on those UKCPVS growth room experiments. What you can see is wherever anything is classified as resistant in the growth room experiments, then by and large we're not seeing any of those varieties getting more than 5% disease in the field. So it's providing good validation of those growth room experiments. But there was one exception, and that's variety 7, and that had more than 5% disease in the Sunderland trial. So that year, that variety was reclassified as being susceptible. But this year, we've piloted a new way of validating those growth room experiments. So that was sowing winter wheat varieties in the spring, so that those varieties would be at the young plant stage when yellow rust pressure is the highest in the field. So but there were 3 trials. There were 2 in Cambridgeshire and 1 in Midlothian. They tested the 21 RL varieties, and they were run by AHDB, NIAB and SRUC. 2 of those trials were naturally inoculated, and 1 was artificially inoculated with yellow rust isolates that were characterised as elsewhere in the UKCPVS project. So I'm just going to take you through the data from those trials, and I've anonymised the varieties throughout these results, but at the end you'll see the reclassifications. So here's the first trial. This was Histon in Cambridgeshire. It's followed spring barley. It was sown on the 13th of May, and it was artificially inoculated on the 29th of May. We've got 2 assessment dates, one on the 13th of June and one on the 21st of June, and we can see a good range in disease incidence. So some of the varieties had no disease, and other varieties had up to 70% disease, and a good spread of disease between them, a good differentiation of the disease at this trial. Our second trial in Cambridgeshire was at March. This followed maize, and it was sown on the 10th of May. And this was naturally inoculated, and it was assessed on the 18th of June. The first thing you'll notice is the disease is much lower in this trial. It was naturally inoculated, whereas the previous trial was artificially inoculated. But nevertheless, we are still getting some good, good. Does that take me back? There we go. Some good differentiation between the varieties, and some good correlation between the previous trials. So the data is just making sense. And then the final trial was at our Easter Howgate site in Midlothian. So this was also naturally inoculated. It followed spring barley, and it was sown on the 14th of May. And here we've got 2 assessments, one on the 20th of June, one on the 4th of July. And again, we're getting a good range of disease, close to 1% in some varieties. Can I go back a slide? Is that all right? And then up to sort of 25% in other varieties. And again, a good differentiation of the disease between the varieties. And then when we mean the data across the sites, again, we've got that nice spread in disease. So we've got the resistant varieties over on the left and the much more susceptible varieties over on the right. We can use this data then to validate what we were seeing in the UKCPVS experiments. And this is where we can do that. It's quite a busy slide, so I'll just take you through it. So we've got the varieties anonymised. What's happened here? There we go. Anonymised on the left. And next to that, we've got the adult plant resistant rating here. And then this column here is the UKCPVS growth room experiment results, so the classification of either resistant or susceptible. These middle 5 columns are the data from the spring sown winter wheat trials, and the final 8 or so columns are data from the RL where disease was collected pre-growth stage 55. So we've got about 5 or 6 RL trials there, with some of them having more than one assessment. So we focus on the RL trials first. We can, if we look across to wherever there were varieties in the growth room experiments were found to be resistant, but finding a good correlation with the RL data. So generally speaking, none of those, well, in fact, all of those varieties that were classified as resistant in the growth room experiments had no more than 5% disease in those RL experiments. And whether they were susceptible, they were getting much higher levels of disease. When we look at the spring sown winter wheat trials in the middle, so the data that I've just shown you, we can see generally we're getting good correlation with the UKCPVS growth room experiments. So generally where there's something that's classified as resistant, it's getting a low level of disease, but there were a couple of exceptions. So varieties 8, 9 and 13 were found to have higher level, more than 5% disease in the field, whereas they were shown to be resistant in the growth room experiments. This information is then assessed by our disease ratings team and also in consultation with the breeders, and then the reclassifications are made. And here we've got the RL classifications of young plant resistance. So last year's RL and the brand new RL that's out, and you can see where the classifications are. An additional change we had this year for classifying something as being susceptible at the young plant stage was that needed to be more than one trial where more than 5% disease was seen. So that could be either the growth room experiments, RL trials, or the spring sown winter wheat trials. Generally speaking, all the varieties retained their classification between last year and this year, the one exception being LG Astronomer, which was classified as resistant last year but downgraded to being susceptible at the young plant stage this year. So just to conclude, the spring sown trial showed that some varieties that were found to be resistant in the growth room experiments were actually susceptible in the field. This probably reflects the limited range of isolates that are used in the growth room experiments and the wider range of isolates seen in the field, and also the differing environmental conditions. But most varieties did retain their classification. There was only that one change. We're going to continue, well, overall, these spring sown trials provide really useful validation of those experiments. And we're going to continue doing them in the new iteration of the UKCPVS. As Kostya was saying, we're going to try and shift the sow dates slightly earlier so that the varieties are probably challenged slightly more in the field, so the cooler conditions are more conducive to the disease. And there was also going to be further emphasis on collecting pre-growth stage 55 data from the RL trials, because it's obviously a fantastic resource for us. So that just leaves me to thank you, and also to thank the ADAS delivery team, NIAB and SRUC delivery teams as well, and answer any questions if you have them. Any burning questions?